Fine, whatever you would want to do is fine anyway. But
You know,
take
but I
care.
respect your privacy in your home and
Appreciate it.
I don't know, it's just my whole.
I've had these kids be like, can I DoorDash lunch? Sure. We care.
I would have gotten you something from Chick-fil-A too as a.
Oh, no,
you know,
we're fine. I seriously, like, I will chow down and.
You get a big lunch later, or like big dinner later, it's just, time bar is my best friend, right?
Oh, yeah, yeah.
Those dark chocolate cherries.
Yeah, those are
The
good.
peanut butter chocolates are good too.
I will like chomp down on a cheese stick or something.
You really.
Cause it's easy.
Protein as well.
Yep.
And the other cheese on Friday.
Friday, yes.
Just sending a little.
To your mom.
I'm like, don't do
Saturday.
If
Yes,
we bring some stuff home, should we bring it home into like the brown?
that's up to you
folder?
guys, however you wanna do it. Hmm. All right.
When they grab me, she put you in there.
Yeah.
Everybody on the 15th?
Evan?
Ready?
That's great, y'all pack it up, time for class.
16.
All right, good job, good start, everyone. Take your pick. I'll take one. I'll take those. Thank you. All right, cool. All right, juniors, y'all are dismissed. See you next week.
I already know who did it.
Oh, you think so, huh?
It was Hope.
No, she strangled herself.
Suicide.
Okay.
Blamed it on the fiance.
Blamed it on the
He didn't
fiance.
like him. He wanted to get locked up.
He's a piece of work, isn't he? Mm-hmm.
So that's always how it ends up in cases like this. It's always some like the thing you like they'll introduce like everybody who like seems so obvious.
There's a twist.
But like it's actually the grandfather of the guy who worked at Arby's.
Yep.
Turns out he actually had some inner motive because like he got his apple stolen in 2019.
Uh-huh.
It's always like that's how like crime shows work. They'll locally introduce the whole thing.
You may. See you later,
Just,
honey.
like tell you actually did it, like Friday. But they'll
Why
like
is it giving me this weird?
give you like absolutely did not do it. I remember I said there was Saturday Live on it. It's like, here's a picture of the victim on a roller coaster. Now he's at a subway station. Now he's surfing. Here's the murder. You already know who it is, but this is an hour-long show, so we're gonna
Stretch it out, huh? Well, this one's going to be a little trickier to just guess. So you're gonna have to work your way through it to figure it out. Hold on over there. There we go.
He's actually the toaster oven.
Toaster oven. You never know. Might be.
You bit her finger and shit over.
Bye, Junior. See you next week. It was you? What? Xander, you been out strangling young girls?
I used a gun.
Officer,
Jeez.
it was not me, but I remember I used a gun. Stringer. What?
Excellent. Anybody have any suspicions yet?
Yes.
Hope and Faith boyfriend.
You think it's James?
It's always the boy.
Because it's always the boyfriend, isn't it? Not James. It is James. Is it James? We don't know. But James is such a nice guy.
He is?
No, he's not.
He's guilty.
He's acting guilty, but he has a cute dog.
Yeah, but that's not the dog.
12 hours, that's 12 hours that he could have been murdered.
Okay, 12 hours that he could have been murdering.
Dog needs a better owner then.
There you go. Yeah, that's true. That poor dog. Mm-hmm.
Hey, bring him. Could just been playing voice chat.
The old lady across, well, hey, could have been Miss Williams.
He also broke up with Hope.
Mr. Mims? That could have been Mr. Mims?
I bet you had something to do with the fact that she was a veterinarian, because it's really specific, and everyone else is like dogs.
Anybody meet Becky's fiance yet?
Yeah.
What'd you think of him?
Why haven't met him?
He's got the haircut.
With the haircut?
Yeah, that's the point I'm looking for.
Luke?
Why didn't they just murder him?
But he's such a quality guy.
AI has some interesting preferences.
Do what? The haircut's too perfect.
I'm so shocked.
Did Bateman have any feedback on him?
It's Batman.
It's Batman.
It's Batman. Did Bateman write anything at the bottom of his interview with Luke? Did anybody see it? What did you do, McKenna?
Is smart enough to commit murder.
Bateman's a little salty, isn't he?
I thought you said Batman. I'm like, what?
Oh, that is his name. It's like a murder Batman. It's Batman.
Batman. All right. So, are they going home? Is anybody leaving him here? Or are they going home? They're going home?
We're going home. After I was splitting them.
All right. Okay. That all works.
Okay, are you thinking Dr. Walker? Are you thinking Dr.
Loki, I'm gonna take it to a police station and ask him for help.
Okay, great. Good job.
They'll have it solved.
What was her job? Did anybody get that?
Veterinarian.
She's not a veterinarian. She's a vet tech. Right. Okay. A vet tech. All right.
This is really cool, by the way.
Thank you. You like it?
AI
I had a lot
guys
of
do-
writing the interviews. Probably that was my favorite part because I had to make everybody into a character. So I had to write a dialogue where people were like character. Like, okay, I'm gonna tell you my favorite is Betsy. What did we notice about Betsy?
She's old.
She's old.
And she doesn't.
She's deaf. She kept asking him to repeat himself, right? Come again. What'd you say? Sorry. And at the end, he said something about Hope's apartment being tidy. And what did she say? Did anybody catch it? I wouldn't call them tiny. She doesn't hear very well. All right, cool. Well, good. I hope you enjoy everybody. They're fun people.
Very good person to be the. If she were to hear anything.
Oh yeah. She's right across the hall.
Person who could hear everything deaf.
Yeah, she's right across the hall. She would have been a great witness, but she doesn't hear too well. But then she claims that she can hear just fine because that's how most old people are. I'm fine. There's nothing wrong with me. Mhm.
You're the one who's speaking.
Yeah, just ask my father who thinks he can still use power tools and in the last 2 weeks has cut his arm and his thumb. 2 sets of stitches. Yes, sir.
Something I did notice is that she said that she didn't hear the dog barking, but someone else heard the dog.
Yeah, she didn't hear the dog barking, but someone else did hear the dog barking. So interesting. Cute little Mr. Mims. All right. So now we're going to start Unit 4, Genetics. We're in Q2. That means your progress reports are going to be coming out very soon. If there's something you haven't finished, you want to finish it real fast so that you're going to get credit for it. If you get late credit, it's better than no credit, right? Zeros are not fun. We should try to avoid them at all costs. Right, people? All right. So let's go. Let's see what we're doing. So we're going to start with Gregor Mendel. Gregor Mendel is one of my favorites. You like him too? He is one of my favorites. Actually, one of my one of my biology professors in college named her Australian Shepherd Mendel, and I just loved him. He was great. Oh, what? An AI generated that? Oh, I did this. These slides have been around for a couple years, so the AI stuff's all new. So we're gonna learn about him. So this is Gregor Johann Mendel, and we're living in, I'm gonna move this up all the way a little bit. So we are in mid-1800s, right? Mid to late 1800s, I guess, really. He's a polymath, which means what? To everybody? Knows a lot of things. So what do we suppose are some of his areas of interest? Caitlin, what do you think?
Psychology.
Psychology, good guess. What do you think, Yusuf? Biology, well, obviously we know that one. Asher? Mathematics. Claire?
What's it called? Philosophy.
Philosophy. All right, Lucas? Physics is a good guess. Barrett?
I'm gonna say botany.
Botany, obviously. Andrew? Music, good guesses. All right, so our buddy Mendel did meteorology, so that's weather, right? Mathematics, biology. And then he was an Augustinian friar and an abbot at St. Thomas Abbey in Brno, Monrovia. Okay, now when Jason and I traveled overseas, we actually drove through what is now Slovakia and past this town, Brno, and had a 30-minute conversation about how in the world you pronounce something that is lacking vowels in the middle of all of the consonants. We're like, is it Barno, Birno, Burno? Is it Bruno, Bruno, Brino? What the heck is it? I actually have a friend whose husband's from the Czech Republic, and I straight up asked her, and it's almost like Bruno. It's almost like Bruno, but with less of a, it's like Bruno. So he was born Johann Mendel, and he was the son of a struggling what? What do you suppose his dad did? What's your guess?
Lawyer.
A lawyer, a struggling lawyer. There are lots of struggling lawyers, Asher. What do you, Yusuf?
Carpenter.
Carpenter's a good guess. He's a farmer, which kind of makes sense because this guy ended up working with plants, right? So in a German-speaking family, in what country do you suppose this is today? What is it?
I'm gonna say Czech. No.
Yes.
Czechoslovakia.
Yeah, Czech Republic. Yes. It used to be Czechoslovakia. It's been lots of things. Now it's Czech Republic. Yeah. Bohemia, basically, right? Czech Republic. And he was given the name Gregor when he joined the Augustinian monks. He became a monk in part because it enabled him to obtain something that he didn't have to pay for. There you go, Claire, education. The monks get educated. And this is great. The monastic life, in his words, spared him the perpetual anxiety about a means of, what do we think? What do you think? Money is a good way to put it. Livelihood is his quote. So some of you will remember, some of you who've been around for a couple of other classes will remember this, but a lot of our great scientific minds in history were either monks, priests, or independently wealthy. Why is that? Why, Asher?
They had access to education. And
That's
food.
big part, access to education and food. What else?
They had a lot of time on their hands.
They had a lot of time on their hands. Okay, if you're a priest or a monk, or you're independently wealthy, chances are you've been educated. Either somebody paid for it if you're independently wealthy, or you get educated by the church, but you've been not just educated in the what of things, but educated to ask the why, right? Guys that go through all of their seminary education get at the why is this included in the canon? Why is this not in the canon? Why, what is meant by this scripture? What is the consensus? And they learn to think deeply. So because of that, they make really good scientists. And what are they not having to do? Priests, monks, and independently wealthy people.
They have fun enough to like go out there to do other.
They don't have to stand out in the heat all day and farm and work and keep shop and do whatever it is they're doing. So because of that, a lot of these guys historically are our good scientists. Yeah.
Which one was Albert Einstein?
Well, that was later. He wasn't any of those things, but he was later. I'm talking like historical. This is pre-Einstein, because if you look at the dates here, he's dead before Einstein really becomes a thing. So yeah. So now we like, I mean, just think about it. There's a dishwasher in your house, probably, right? There's a washing machine in your house, right? These are things that save you time. So you can do work and save time differently. But in the past, you didn't have a dishwasher or a clothes washer. You're doing that all by hand. Everything took more time. Everything did. So Mendel gained posthumous recognition. What's posthumous mean? After death. Yes, posthumous recognition as a founder of modern science of what, Eunice? Genetics. Very good. So his pea plant experiments were conducted. Here are dates I'll show you between, come on, I'm clicking. My mouse is being so weird today. Oh, don't do this to me. There we go. Between 1856 and 1863. And they established the rules of heredity, and we now call these the laws of what, do you suppose? Does anybody know? These are the laws of Mendelian genetics. So remember, his name was Johann. He became a monk. They gave him the name Gregor, and which is basically Gregory, right? Saint Gregory, that's who we're dealing with here. And then now we call this Mendelian inheritance, which is your most basic understanding of how inheritance and genetic traits work. it's far more complicated than what he discovered, but he kick-started this, and he's our father of modern genetics. So he coined two terms about traits. What are traits? They're either one of two things. What do you think?
Quantitative qual-
Quantitative and qualitative is a good guess. That's a good guess. Yeah, what do you got?
dominant, recessive.
There's the ones. Okay, dominant and recessive. So he coined these terms, dominant and recessive, in reference to traits. And he published his work in 1866, demonstrating the actions of invisible factors. We now call them what? Say it.
Gene.
Genes. That's what determines our trait. But they were invisible factors. So the profound significance of his work wasn't recognized until the turn of the 20th century. So a good 3 decades after he was gone from the face of the earth. Poor guy never got his credit in his lifetime, which is very sad. It was basically a rediscovery of his laws, and it ushered in the modern day. Huh?
Some Franklin.
Oh yeah, another one. All right, so let's look at this pea blossom for a minute first, because you have to kind of understand the structure of a pea blossom to understand what he was doing. So he worked with pea plants, because he could very much control what he was working with, and he worked with purebred pea plants. So here's your pea plant. Now, a pea flower, a pea blossom, is going to have male and female structures. Does anybody know which are the male structures in a flower in general? Do you know? Barrett, do you know?
I wanna say the stigma is the male and the.
Good guess. Partly right. Do you know?
Stigma and ovule.
Partly right. So sort of. All right. So your male parts on a blossom are going to be your anther and your filament, and then your female parts are all the rest of those other structures. So how it works is, you've looked and seen filaments in flowers, right? Like picture an azalea, you see the little things, they stick out, right? And at the top, they have those little bulbs at the top. Those are your anthers. That's where you find pollen, right? That's where your pollen is. Your pollen is your male gamete. It's your male sex cell. It's your sperm, essentially, right? And then your female part is going to be your pistil, which is composed of your stigma, your style, your ovary, and then your ovules down at the bottom. Now, if this were an apple blossom, all right, you would have multiple ovules in there, and the ovary would turn into the apple, and all the ovules would turn into the apple seeds. Okay, but we're talking about a pea, so it's just one seed, right? So this is going to be your fruit here. This is going to be your pea eventually, or your pod that makes your peas, really. So this is what we got. All right. Male and female parts. So that little stamen has that pollen on it, and if that pollen lands on the top of the pistil there at the stigma, there's a little hole, and it will actually fall all the way down in there, and that's how it fertilizes the ovule. So what is the name for the process where new cells are formed from a male and a female gamete? Male and female gamete come together, Caitlin?
Fertilization.
Fertilization is the word we are looking for. There you go. So you have fertilization, and in flowering plants, you have two types: self-pollination, cross-pollination. Self-pollination, the anther produces pollen, the pollen gets on the pistil, goes down, and the flower has fertilized itself. Is that flower is the offspring going to be genetically identical or different from the parent?
Identical.
Identical, good. Over here you have cross-pollination. So you have two things. So this can be done by wind, by insects, hummingbirds, butterflies, bees, these types of things. Huh?
And a paintbrush.
Yeah, and a paintbrush, which is what Mendel did. But you're going to take the male gamete from one flower and move it to the female gamete of another flower. So this is how you cross-pollinate. So what Mendel did is he recognized that pea plants are usually self-pollinating, which means pollen from a flower lands on the pistil of the same flower. Whoop, why are you doing that? Stop. There you go. But he created a method where he could control the cross-pollination of pea plants. So he was breeding pea plants with the purpose of cross-pollinating them, and he could control it. So what he did is he would take the anthers from one blossom and cut them off, and then he would take the pollen from those anthers and move it over here to a different plant, and that one go to this one, and this one go to that one. But he cut those anthers off so that it wouldn't self-pollinate, right? So he removed it from the equation, basically. And he used a little tiny paintbrush when he did it. So a purebred organism is an offspring of many generations that have the same traits. So these peas are purebred. So it's been this breed of pea with this breed of pea forever. And he started his experiments with these purebred plants. So he studied 7 traits, and these are the 7 traits he studied. Did you know peas had this much variety, for goodness sakes? He studied whether the seed was round or wrinkled. Nobody wants to eat the wrinkled ones, right? Probably not. Fabulous. And whether the cotyledon was yellow or green. Now, if you open up a pea, it'll split into 2 pieces, right? That's how you have split pea soup, splits into in half. That means it's a dicot, and we talk more about plants later. We have dicots and monocots. Peas are the cotyledon, which is the part is like, remember when we did our little beans that are still growing over there? When it opened up, those are the cotyledon. And some organisms, some plants have 1, some plants have 2. So is whether the cotyledon is yellow or green. He studied whether the flowers were white or violet. He studied whether the pod was full or wrinkled up and constricted. He studied whether the pod was green or yellow. And then where it says place here, it's that axial pods and terminal pods. Can you tell what the difference is there? Can you tell, Barrett?
Yes. One of them has like blossomed, and one isn't.
They both got blossoms. There's the one at the top and one at the bottom. Can you tell what the difference is, Ridley?
One is closer to the base of the stems.
Yes.
One is at the top.
Yes. So what it is axial pods, the top one there, grow along the stem. Terminal pods grow at the end of the stem, at the terminus of the stem. So that's the difference between axial and terminal. And you'll find that if you use like a dichotomous tool or whatever to identify plants, it'll ask you if the leaves are axial or terminal. All of that's different. Okay, and then size, short and tall. Now, in human beings, height is not determined by one gene. It's a combination of genes and obviously environmental factors. If we all stood in a line in here, we could probably put ourselves by height, shortest to tallest, but we're not all 6 foot or 5 foot, right? We're different. We're not all tall and short. We're a spectrum of height. But in pea plants, it literally is one gene that determines it. Either they're tall or they're short. So he used these 7 traits. And here's what he did with them. Here's a cute little TEDx. Look at the cute peas. They're happy peas. Look at them. Play.
These days, scientists know how you inherit characteristics from your parents. They're able to calculate probabilities of having a specific trait or getting a genetic disease according to the information they have from the parents and the family history. But how is this possible? To understand how traits pass from one living being to its descendants, we need to go back in time to the 19th century and a man named Gregor Mendel. Mendel was an Austrian monk and biologist who loved to work with plants. By breeding from pea plants he was growing in the monastery's garden, he discovered the principles that rule heredity. In one of the most classic examples, Mendel combined a purebred yellow-seeded plant with a purebred green-seeded plant, and he got only yellow seeds. He called the yellow color trait the dominant one, because it was expressed in all the new seeds. Then he let the new yellow-seeded hybrid plants self-fertilize, and in this second generation he got both yellow and green seeds, which meant that the green trait had been hidden by the dominant yellow. He called this hidden trait the recessive trait. From those results, Mendel inferred that each trait depends on a pair of factors, one of them coming from the mother and the other from the father. Now we know that these factors are called alleles and represent the different variations of a gene. Depending on which type of allele Mendel found in each seed, we can have what we call a homozygous pea, where both alleles are identical, and what we call a heterozygous pea, when the 2 alleles are different. This combination of alleles is known as genotype, and its result, being yellow or green, is called phenotype. To clearly visualize how alleles are distributed amongst descendants, we can use a diagram called the Punnett square. You just place the different alleles on both axes, and then you figure out the possible combinations. Let's look at Mendel's peas, for example. Let's write the dominant yellow allele as an uppercase Y, and the recessive green allele as a lowercase y. The uppercase y always overpowers his lowercase friend, so the only time you get green babies is if you have 2 lowercase y's. In Mendel's first generation, the yellow homozygous pea mom will give each pea kid a yellow dominant allele, and the green homozygous pea dad will give a green recessive allele, so all the pea kids will be yellow heterozygous. Then, in the second generation, where the 2 heterozygous kids marry, their babies could have any of the 3 possible genotypes, showing the 2 possible phenotypes in a 3-to-1 proportion. But even peas have a lot of characteristics. For example, besides being yellow or green, peas may be round or wrinkled. So we could have all these possible combinations: round yellow peas, round green peas, wrinkled yellow peas, and wrinkled green peas. To calculate the proportions for each genotype and phenotype, we can use a Punnett square too. Of course, this will make it a little more complex. And lots of things are more complicated than peas, like, say, people. These days, scientists know a lot more about genetics and heredity, and there are many other ways in which some characteristics are inherited, but it all started with Mendel and his peas.
So these are our basics, right? Our very basics. All right, so genes pass what? What's the word we use for characteristics when we're talking about genes? Lucas.
Traits.
Traits. They pass traits from parent to offspring. And what do we call different forms of a trait?
Alleles.
Alleles. Okay. That's a fun word. Some people have trouble with that one. Alleles. All right. Now, in its most basic form, Mendelian genetics, each trait is derived from, yes, Eunice. Well, how many alleles? That's what we want to know. How many alleles? Two, right? So a pair. All right. And each allele is categorized as either dominant or recessive. Now, dominant alleles do what to the recessive alleles? There's a word here we want, you know? Cover is the word, Lucas. Very good. Cover. This is the word you need to know. If you have a dominant allele and you have a recessive allele, the dominant allele is always going to cover the recessive allele. That genetic trait is there, but it is covered by the dominant allele. Now we'll learn in the next couple of weeks that sometimes there are things where like one gene will switch on and the other will switch off, and that's how you get things like cats with tortoiseshell pattern. It's really kind of fascinating. But anyway, it is definitely the case that your dominant is typically going to always cover your recessive. Cover's the word you want to use. So what you call a combination of alleles then, two alleles together, is a genotype. And then the expression of those two alleles is called what? Lucas? It's a phenotype. All right. So your genotype is what your genes are. Your phenotype is how it's expressed. So you can kind of think of it as what it looks like, but it's not always what it looks like because sometimes you don't see it. But it's the product. It's what is expressed in the end. And remember, the recessive allele is not going to be expressed if a dominant allele is present. The only way the recessive allele is expressed is if there is no dominant, so you have two recessive alleles. That's the only way you're going to get the recessive allele. So look, it's that recessive phenotype. So look here, we have cherries. All right, what do you suppose is the dominant color phenotype for cherries, McKenna? It is red, because that's a capital letter. That means you know it's going to be red. Now, if you have two capital letters, two dominant alleles, your phenotype is going to be what, everybody?
Yellow.
No, right here. Two dominant, capital letters,
Red.
red. All right, now over here, you have a dominant and a recessive, but what's your color? Red. Red phenotype because the dominant is doing what? Covering. Very good. And you've got two lowercase, so you have recessive here, so your recessive color phenotype is what? Yellow. Got it. You've got it. Very good. So your dominant alleles are designated with what kind of a letter? Everybody, capital. And your recessive alleles are expressed with what kind of a letter? Lowercase. We're good. Look at us. We're so good. All right. So this is the principle of dominance. And it states that that dominance always exhibit that form of the trait. If it's dominant, it's always exhibited. So either as a capital A, little a, or two capital A's. And that the recessive exhibits the trait only when a dominant is not present. So now we're going to talk about hairless guinea pigs with the amoeba sisters for a minute. Okay. You ever seen a hairless guinea pig? I think they are atrocious, but some people like them.
One card, two ways.
They're funky looking little things.
No credit impact to apply.
Yes, commercials, always. Oh, come on.
There's a reason ball boys make especially attentive waiters.
Yes, yes. There we go. Love commercials. So fun.
So I have a confession to make. For my classroom, I always wanted a hairless guinea pig. They sell them at some pet stores, but they can be really hard to find. And while I doubt they do so well in the wild, their genetics are fascinating. And so are guinea pigs. We spend quite some time on LOL cats, but we'd spend even more time if someone would make a funny guinea pig site. Most guinea pigs have hair. This is because of their DNA. DNA makes up genes, and the guinea pigs received genes from both their mother and their father. An allele is a form of a gene and often represented by a letter. In this case, we'll use the letter H for hair. A hairless guinea pig has two recessive alleles for the trait of having or not having hair. A recessive allele is usually represented by a lowercase letter or allele. By recessive, it means that the allele will not usually show up. The only way it will show up is if there is no dominant allele present. A dominant allele is represented by a capital letter, and it's an allele that will show up. Think of it as dominating. So a hairless guinea pig does not have a dominant allele for hair, and that's why the recessive trait, not having hair, shows up. A hairless guinea pig's genotype, that's the genetic makeup of an organism, can be represented as a lowercase little h. A guinea pig that does have hair can be represented as a big H, or a big H, little h. See, it only takes one dominant allele, a capital H, for a trait to show up. That dominating allele means that the recessive allele is hidden. Now, we're not quite done with the vocab here. A genotype of big H, or little h is considered homozygous. The root in this word means same, and they are the same case. Big H, those are both capital letters, and little h, those are both lowercase letters. So big H is called homozygous dominant because of the capitals, and little h is homozygous recessive because of the lowercase. A genotype of big H, little h is considered heterozygous. The root in this word means different because their letters are different cases. One is a capital and one is lowercase. If I were to show you a guinea pig with hair, would you know its genotype?
Would you?
Well, you would know it's not little h, because if it was little h, then it would be hairless. But remember, a guinea pig with hair, it could be big H, or big H, little h. We don't really know. We could do a test cross to eventually determine this, but you can't tell just by looking at the guinea pig. Now, if we had a hairless guinea pig, we do know for sure that it's little h, because if it did have a capital letter, even one, that dominating allele would take over and it would have hair. So let's try a monohybrid cross. The root mono means one because it focuses on one trait, in this case, hair. And that means a Punnett square would need to be created like this with 4 squares. In our cross, we are going to cross 2 heterozygous guinea pigs. So let's look at this in steps. Step 1, first, figure out the genotypes of the parents. Heterozygous means big H, little h. Step 2, place one parent along the top of the Punnett square, outside of the boxes like this, and place the other parent along the left side of the square, outside of the boxes like this. Step 3, cross them. For formatting purposes, we always put the capital letter first. The results that you get in the squares would be the offspring, the babies. Now, if I were to ask you the genotypes of the babies, you could list them out. 1 big H, 2 big H, and 1 little h. I could even turn that into a genotype ratio of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive, or a percentage. 25% are homozygous dominant, 50% are heterozygous, and 25% are homozygous recessive. Well, what if I asked about the phenotypes? What does that mean? I like to think that pheno sounds like physical. So the phenotype would be the physical traits of that organism. And in this example, it's whether they have hair or not. And remember, any babies that have a capital H have a dominating allele, and they will have hair. So the babies that are big H, or big H, they have hair. So 3 of them here, the 1 big H baby and the 2 big H babies, they all have hair. The homozygous recessive baby, which was little h, that baby has no dominant allele present. So this little guy would be hairless. So you could say the phenotype ratio is 3 have hair to 1 that's hairless. Or a phenotype percentage could be 75% hair, 25% hairless. One thing to emphasize about Punnett squares is that they are predictions. These are probabilities. This means it's not necessarily exactly what you're going to get. For example, it's a probability that a child has a 50-50 chance of being born a boy or a girl. But we all know families that only have children that are girls, or families that only have children that are boys. Probabilities are predictions. Another reason to love biology, it's exciting. Well, that's it for the Amoeba Sisters. I'm gonna remind you.
So I can predict based on that something about a family member that we have. So if I look at my husband's family, his dad has blue eyes, his mom has brown eyes. Both my husband has blue eyes and his sister has brown eyes. Brown is dominant. What must my mother-in-law's genotype be? What's her phenotype? She has brown eyes. She has brown eyes. Her husband has blue eyes. She has both brown and blue-eyed kids.
You have the dominant recessive gene.
Right. She has to be heterozygous dominant. She has to be heterozygous because she has to have the recessive allele that produces blue eyes, or my husband would not be blue-eyed. So I know that about her without ever having looked at her genes. I know that must be her genotype. If she had all brown-eyed kids, there could be probability she could still have that recessive allele and it just didn't show up. But because there's a blue-eyed kid, I know she has to have a heterozygous genotype for eye color. And eye color is also, there's codominance involved, like green eyes are a whole different thing, so anyway, how do we apply these principles? So just so you know, this is Mendel's garden when he was working in it. It's Mendel's garden now. Those look like begonias, not peas, but hey. So you can visit it, should you care to go. It's in Monrovia now. You could go there and you could visit it. So let's have a look at some of how this works. So for example, here's for our pea plants, tall and short. Capital T is your dominant trait for tall. Lowercase t is your short recessive trait. So your genotype, what do we call this genotype with the capital T, capital T? What's it called? Homozygous dominant. Okay, so everybody, what do we call the middle one here?
Homozygous recessive.
Homozygous recessive, and the bottom one is going to be heterozygous, which is going to be the dominant expression, right? So we get, my mouse is giving me trouble today, and it's making me insane. There we go. Maybe not. I'm clicking you. Click. Oh, man. Let me if I just tap the screen. Oh, that's fun. I didn't want it to do that. Hold on. It's coming. It's just here we go. Maybe not. Okay. Homozygous dominant, like you said. Homozygous recessive, if it will click. Here you go. All right. Now let's look at phenotype. What's the phenotype going to be for this first plant? Tall or short, everybody? Tall. And how about the next plant? Short. And how about the bottom plant? Tall. You've got it. All right. Now, chromosomes exist in pairs.
Can I use a sheet paper?
Oh, you want some paper?
Can I have a sheet paper?
Yeah. Did
Alright.
you see what I saw, like the brownie point?
Yeah, there you go. Okay, cool. All right. So, caught a brownie point. Somebody explain to Macrina what are brownie points. Tell her.
When we find a mistake in one of the lessons, we write it down and put it over there. And if we get, was it
25.
we get brownies.
There you go. Catch my mistakes. So chromosomes exist in pairs. You can think of them like shoes. So if you look at these shoes here, you can probably match these pairs, right? You can look at these and go, all right, if I found this pile of scattered shoes, I could match them and put them with their correct pair. So we call them homologous pairs. So look at your shoes, like for real. If they're still on your feet, look at your shoes. Now let me ask you a question. Are your shoes identical? Really? Can you put your right on your left? Oh, wait. So they're not identical. You have a right and a left.
Mirror.
They're mirror. Yeah, they're kind of mirror. But so they're a pair and they go together. And if you put them side by side, like I can see the stitching on my boots is the same on both. They line up. If you've got laces, you probably notice your laces line up, right? If you're wearing shoes that match, that is, right? And then, but they're a pair, but they're not identical. So this is how homologous chromosomes work. Pair, but not identical. They're sisters. And it's not clicking again. Oh my goodness, this is going to make me crazy. Go. Similar, but not identical. You have a left and a right, for example. Now, if we took all the shoes in class and threw them into a big pile, would you be able to sort them back into their pairs?
Yes.
You would, right? Okay. It's because no two pairs are identical, right? So all the shoes are in class are kind of like your unique pairs of homologous chromosomes and cells. Yes.
What if people wearing the same-
That would be a problem, but they still wouldn't be identical because I guarantee you the wear patterns would be different. The size might be different. Yeah. The way you walk, you wear the bottom of your shoes differently, right? All right. So chromosomes exist in pairs. So you've probably seen chromosomes shown looking kind of like this, right? And that's because they exist in pairs. So they match, but they aren't identical, just like shoes. So if we look, we're going to consider this chromatid. Each one of these is a chromatid. Together, they're a chromosome. But remember, we count centromeres. So if you're counting them, you can count them. Anyway, let's just say blue and pink, even though they're both look like shades of purple to me. The blue ones are your paternal copies, so they came from your father. And your pink ones are going to come from your female parent. So if you look here, do you notice that the banding is the same? That's how genes are on chromosomes. They're banded like that. They're parallel. They're across from one another. And that's why they're paired. But as you'll see in a second, you can have different alleles that create that gene. So if you look right here, here's your blue eye, brown eye example. You have an allele for blue eyes on this chromatid and an allele for brown eyes on that chromatid, but it's still the allele for eye color, the gene for eye color that's parallel. You see it, right? So each chromatid has a series of alleles, and its sister chromatid possesses the same series in the same order, so that you get pairs within pairs. Does that make sense? Pairs within pairs. So during meiosis, the process of gamete production, we're going to get what we call chromosomal segregation that allows for independent assortment of genes by the production of gametes with different combinations of chromatids to produce all of our varying traits. What was the name of the process by which a cell divides and makes daughter cells of itself? We did that.
Mitosis.
Mitosis, right. That's mitosis. This is meiosis. Isn't that fun? They have to be so similar. So mitosis is basically how cells generate new versions of themselves. And are those cells genetically identical or genetically different? What are they? They're genetically identical. With meiosis, what you're producing are gametes. Gametes are sex cells, so sperm or egg, and they are going to be what we call haploid. Haploid meaning half. It has half the chromosomes it started with. How many chromosomes do human beings have?
46.
46. So how many are going to be in a haploid cell?
23.
23. Very good. So all the sperm, all the eggs have 23 chromosomes. And when those chromosomes come together, you got these chromatids, bleep. Now you have a new genetic makeup because that's how those genes are going to combine. Did you have a question, Eunice?
The question isn't meiosis an eye part too?
Is meiosis an eye part?
I searched it up and then it's something related to the eye.
I'll have to look that up. I'm not sure what you found there. But so meiosis is your gamete production process. So we have Mendelian laws. All right, we have a principle that predicts that each phenotype is determined by two alleles, one of which is present, known as the dominant allele, and the phenotypes are expressed only when an organism is homozygous recessive. Which law is this? Do we know? Does anybody know? That's the principle of dominance. And this is what we just talked about with the little cherries. All right, now we have this principle that predicts that during gamete production, each gamete receives exactly one of the two alleles. And that's what?
Segregation.
Yes, principle of segregation. If you look down here at the bottom, you see that those chromatids have been separated out. So it's segregation. Then we have this one. Yeah, I know you're seeing them. They go by quickly. Caitlin, what is this one? independent assortment, and it predicts that alleles coding for different phenotypes are segregated independently from one another during gamete formation. And this is how we get so much genetic variety with sexual reproduction as opposed to asexual reproduction, where you get no genetic variety unless there's a mutation, right? Which we'll talk more about later. We're not into mutations yet. Come on, mouse. Mouse is so not fun. So now we're going to talk about meiosis. We just did mitosis last couple weeks. Now we're doing meiosis. Meiosis. So, meiosis is the process by which sex cells or what? What's the word for sex cells?
Gametes.
Yes, good, Lucas. Gametes. Gametes are produced. Okay. We can use that word from now on. It's shorter than saying sex cells or sperm and egg or sperm and ova or whatever. We will just say gametes. So in humans, obviously we have sperm and ovum, singular, ova, woo, plural. That's bouncing around really fast. There we go. So this Amoeba Sisters again, they're going to show you a good graphic of meiosis if my mouse will function. There it goes. Here it goes. We'll probably get an ad. Yep. Hey, choose debit. Pay later.
No credit impact to apply.
Okay.
You are even prettier in person.
All right.
There's a reason ball boys make especially attentive waiters.
Oh, come on. Go. Go. There we go.
Have you ever wondered how two siblings can have the same mom and dad and still look so different? Well, today we're going to talk about a process that makes that possible, a process called meiosis. Not to be confused with mitosis, which sounds unfortunately similar. Mitosis makes identical body cells, like your skin cells and stomach cells. Recall from our mitosis clip that since it makes identical body cells, mitosis is important for growth and for repair of damage, or to replace worn out cells. But not meiosis. Meiosis is a process that contributes to genetic variety. Meiosis also doesn't make body cells. It makes sperm and egg cells, otherwise known as gametes, the fancier word. You might recall that humans have 46 chromosomes. That's how many chromosomes most body cells in your body have. But there are some human cells that don't have 46 chromosomes. Human sperm cells and egg cells have 23 chromosomes. Why the number difference? Well, if a sperm cell has 23 chromosomes and an egg cell has 23 chromosomes, when they come together, that makes 46 chromosomes. That will allow a newly formed fertilized egg to develop into a human. Meiosis is what we call a reduction division because you have a starting cell that has 46 chromosomes, and your ending cell, the sperm and egg cells, have only 23 chromosomes. Before we can start getting into the stages of meiosis to make gametes, we have to remember what happens before meiosis can even start. Actually, this also happens before mitosis. It's the stage known as interphase. If you remember interphase, it's when the cell is growing, it's replicating its DNA, it's carrying out cell processes, and just like mitosis, interphase happens before meiosis is even going to start. So the starting cell has 46 chromosomes, and you have to duplicate those chromosomes in interphase before meiosis starts. That basically means you are duplicating your DNA since chromosomes are made of DNA and protein. Ready for the tricky part? Well, because we tend to count chromosomes by the number of centromeres present, when the 46 chromosomes duplicate, we still say there are 46 chromosomes because the sister chromatids are still attached and we're actually counting by centromeres. So 46 chromosomes here, they replicate in interphase, and you still have 46 chromosomes in this picture. But you went from 46 to 92 chromatids. Little tricky there. We have a detailed video that explains these chromosome numbers before and after replicating in interphase that can be useful for meiosis. Okay, so interphase checklist done. Now we can move into meiosis. You might remember the mitosis stages PMAT. The P was for prophase, the M was for metaphase, the A for anaphase, and the T for telophase. And the good news is that in meiosis, you still use those terms. But because meiosis is actually a reduction division, you're going from 46 chromosomes to 23, which means you actually divide twice. So instead of mitosis, where you divide one time and do PMAT one time, in meiosis, you're going to divide twice and therefore do PMAT twice. And because of this, in meiosis, you put numbers after the phases to indicate whether you're in the first division or the second division. So let's dive right in. So let's start with the very first step, prophase 1. One thing I like to remember about prophase is this actually means before, and it kind of helps you remember that it comes before all the other stages start. This is where the chromosomes are going to condense and thicken. They are actually going to line up with their homologous pairs. The word homologous means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. They're going to match up. It is during this prophase 1 that this amazing process occurs called crossing over. Now, I know crossing over probably sounds like something very different, but this is actually a really awesome process because this is when the chromosomes, they're lined up in homologous pairs, and they have this way that they can transfer their genetic information and exchange it between each other. It's kind of like these chromosomes flop over each other and they do a little genetic information exchange here. It makes for what we call recombinant chromosomes, which can eventually contribute to the variety that we were mentioning that siblings can have even when they have the same parents. More about that later. Now we move into metaphase 1. In metaphase 1, think of the M as standing for middle. The chromosomes are now going to be in the middle of the cell. It's a little bit different though from mitosis because these chromosomes are going to be in pairs in the middle of the cell. So it's not a single file line, they're in pairs in the middle. Now during anaphase 1, think of A for away because the chromosomes are going to be pulled away by the spindle fibers. Then we end with telophase 1, where you have two newly formed nuclei, and it becomes obvious that you're going to end meiosis 1 with two new cells. Cytokinesis follows with splitting the cytoplasm, but we're not done yet. On to meiosis 2. The very first step in meiosis 2 is prophase 2. It's not going to be nearly as eventful as it was in prophase 1, though, because they're not going to have homologous pairs of chromosomes. They also are not going to have this amazing process called crossing over. That doesn't happen again in prophase 2. You have your chromosomes, and the spindles are starting to form like they did in prophase 1, but prophase 2 is just not as eventful as having that process of crossing over. In metaphase 2, remember, think M for middle, the chromosomes are going to line up in the middle. This time, though, they are in a single file line. They are not in pairs like they were in metaphase 1. Anaphase 2, remember A for away, but this time it's the chromatids that are going to be pulled away by the spindle fibers. In telophase 2, you can see the nuclei reforming, and you can also see that the two cells have divided. There's going to be 4 cells forming. Cytokinesis will follow to completely split the cytoplasm. Now, keep in mind that meiosis in males produces sperm cells, and in females it produces egg cells. Because of independent assortment and also crossing over, you're going to have variety. For example, in a male, the 4 sperm cells that are produced each time, they are all different from each other. They're also different from the starting cell because the starting cell had 46 chromosomes, and the ending cells only have 23. So they are not identical to the original, and they are not identical to each other. This is going to lead to variety, a reason why 2 siblings with the same parents can look different from each other. They still developed from a unique egg and a unique sperm cell that came together. One last thing to think about. Scientists are often looking into the process of meiosis because sometimes the chromosomes don't separate correctly. It's called non-disjunction when a cell can receive too many or too few chromosomes in the separation. This contributes to some genetic disorders, which is something scientists continue to study. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Does anybody offhand know of a genetic disorder that involves too many or too few chromosomes? Do you know, Kenna? Down syndrome. Do you know which pair that is? 21. Trisomy 21. You end up with 3 chromosomes in what is supposed to be your 21st pair. You end up with 3, not 2. So yeah, that's probably your most common one. There's others as well. All right, so let's have a look at Gregor Mendel's patterns of inheritance a little more. So we also have something called incomplete dominance. Okay, so this is when it gets a little more complicated. So sometimes both alleles at a gene locus, are a locus meaning that's that particular gene, are partially expressed, often resulting in an intermediate or a different phenotype. And what do we call this? Anybody know? Caitlin, do you know? So it's fine. It can be called incomplete or partial dominance. So for example, in roses, the allele for red color is dominant over the allele for white, but with the heterozygous flowers in both, you get both alleles expressed to some degree, so you end up with a pink flower. So now things are getting a little more complex, right? And this is not the case with every gene, it's the case with some genes. And not every organism, some organisms. So what's the relationship between two versions of a gene? Do we know, Yusuf? Codominance. All right, so codominance is a relationship, between two versions of a gene where individuals receive one version of a gene called an allele from each parent. If the alleles are different, the dominant allele will usually be expressed while the effect of the other allele, the recessive one, is covered or masked, right? So here's an example. You can have a white cow and a red cow, and then you can get what's called a roan cow.
For the previous one, for the incomplete dominant, so is it a 50-50 pink and white, or is it like more towards the capital, or is it more?
It really it's going to differ by species and by particular trait. So it's not always going to be exactly the same rule across the board for everything. So in some species, you're going to get more of this and less of that, and then in individuals too. So it just adds to the variety basically, but it's not as straightforward.
It's not always tilted to the capital.
No, not always. I mean, you can see, like, you've seen some flowers that have like even on a bush itself, you might see flowers with varying shades of color, right? It can vary a whole lot. Sorry, now if this would just go, I don't understand why it's so being weird today. All right, so incomplete dominance versus codominance when we compare them. Okay, so if they're both equally expressed and then you get red and white flowers, what do you suppose this one is? Incomplete or codominance, Ridley? She says incomplete, and I'm going to try and make it click. And I'm going to try and make it, there it goes. That one's co. All right, so we get codominance for that one. And then obviously this is going to be incomplete dominance when you have alleles that blend together. So in this case, they're both dominant. They're co-dominant, so they're kind of fighting for what's being produced. And then in this case, you've got both of them together and they kind of blend and you get the pink. So typically there are only two alleles for a gene in a diploid. Diploid meaning it has all of its chromosomes. Haploid meaning it's half, like a gamete is haploid. When there is a gene existing with more than two alleles, this condition is referred to as multiple alleles. So here are different phenotypes and different genotypes for rabbits. You can have brown ones, chinchilla ones, Himalayan ones, and albino ones. And these are all different types of alleles that exist in the population. So it's not just two, it's more than two alleles. So you have different options that can come together. A good example in humans is blood type.
There's still codominance, or is there only one dominant?
It depends on the allele. It depends on the gene. Again, some are going to have codominance and some are not going to have codominance. Some are going to be very Mendelian and be dominant recessive. So again, it's going to depend. It gets very complex. So if we look here, this shows how blood types work in humans, which is a great example of multiple alleles. And we get another ad.
No dreaded impact to apply.
You know. Do we not love Amoeba Sisters? We do. I love it.
There's a lot of phenotypes that are easy to tell. Your eye color, your hair texture, your height, whether you have a straight thumb or hitchhiker thumb. But one phenotype that you can't tell just by looking is your blood type. Your blood is really made up of many things: platelets, plasma, red blood cells. But you've probably heard before that when blood is donated, it's very important that it is matched correctly. And that's true because blood type phenotypes vary. It really boils down to the fact that red blood cells, they're not naked. They have proteins on their surface. And it turns out that your immune system is very protective. And if it gets blood that's donated that has different proteins that it's not used to, it will attack them. With blood type, you can have several different phenotypes: A, B, AB, or O. These letters stand for antigens that are found on the red blood cells. So type A blood, for example, has A antigens on the surface of red blood cells. Type B blood, for example, has B antigens on the surface of red blood cells. Type AB has both A and B antigens on the surface of red blood cells. Type O, I like to think of it looking like a zero. it doesn't have A or B antigens. It's naked. Well, okay, it does have other proteins on its surface, but not A or B. So think of O as looking like a 0, doesn't have A or B. So if you have type B blood, you have B antigens on the surface of red blood cells. That means a person with type B can accept another person's type B blood because B is an antigen that their body recognizes. But if you try to give that person a type A blood type, that's an antigen that the immune system does not recognize. That person's immune system would attack. It would also attack AB blood because that includes the A that it doesn't recognize. Now, type O would be safe. Remember, O, think of like a 0. It doesn't have A or B antigens, so type O can donate to everyone. Now, while O individuals can donate to everyone, they can only receive blood from another type O. Because type O doesn't have A or B antigens, so their immune system will attack any other blood type that does. Neither of us have type AB blood, but we would think this is a cool blood type to have in the sense that you could receive blood from anyone. If an AB person received blood from a person that had type A, well, they've got the A, so it's all good. And if an AB person received blood from a person that had type B, well, they've got the B antigen too, so it's all good. They can receive blood from type O too because there's not any antigens to even worry about. Now, one thing we do want to add that makes all of this a bit more complicated. Blood types also have a plus or minus sign listed by the blood type. This makes a big difference with blood donations. If you have a plus, it means that you have this other little protein called Rh factor on the surface of your blood cells. And if you have a negative, it means that you do not have this little protein called Rh factor on the surface of your red blood cells. Now, we're not going to be able to go into that in this short clip, so to the Google for that if you would like to learn more about this. Blood type is genetically inherited, and a great example of multiple alleles. Remember that alleles are a form of a gene, like a flavor. Just an analogy. So let's put this into practice. Let's say a couple gives birth to a baby boy. Both parents have type A blood, but then there was a mix-up at the hospital. And now, sorry, I get really into my drama. There are these 2 baby boys, and the hospital does not know which one belongs to the couple. Probably not a hospital where you want to have kids, but let's try and use our blood type genetic problem solving skills to help us out here. Baby Phil has type B blood, and baby Sylvester has type O blood. Could either of these babies belong to the couple who both have type A blood? Well, let's talk about phenotypes and genotypes. The phenotype of type A blood is A, but the genotype is written like this or this. The format of writing here helps with multiple alleles like blood type problems, and we can show you why when we work this out in the Punnett square. Now, you may notice that I said that blood type A could be written this way or this way. Without testing, we don't really know which one it is. You could consider this one to be homozygous and this one to be heterozygous. The phenotype of type B blood is B, but the genotype is written as this or this. The phenotype of type AB blood is AB, and the genotype is written this way. There's no other way to write that one. The phenotype for type O blood is O, and the genotype is written like this. Remember how I said that the O kind of looks like a 0, and so you can think of it as having 0 blood type antigens? Well, that's what it is. Little I, no coefficient. Okay, back to the babies. We are told the parents both have type A, but remember, we don't know whether that means they are this type A, the homozygous, or this type A, the heterozygous. And the mom could be one of those genotypes, and the father could be the other genotype, or they could both be the same genotype. We just don't know. Okay, so let's try it out with all of the possibilities. Remember that we write the parent genotypes on the top and the side of the Punnett squares, like this. We'll fill in the Punnett squares, and we'll make sure to have capital I's first, just for formatting.
See the answer?
Okay, so is it possible to get baby Phil, who has type B, in any of these offspring?
Is it?
No.
He must be someone else's baby. What about baby Sylvester with his type O?
Yeah, you see it?
Yes, it is possible,
Right
but
there.
both parents would have to be heterozygous A. That means they both must be this. then yes, you would have a 25% chance, that's 1 in 4 here, of having a baby with type O blood. Of course, if I was advising this couple, I would also insist on a DNA test, as this blood type problem only shows that it's possible that baby Sylvester is theirs. Well, that's it for the Amoeba Sisters, but I remind you to stay curious.
Anybody know their blood type?
Yeah, AP's positive.
AB positive?
Mine is B.
Yours is B? Anybody else?
Positive.
A positive? I'm O positive.
AP's no positive.
I'm all alone. I got no antigens. Do you know yours?
I don't know my blood type, but my friend's mom has RH negative blood type.
Ah.
And apparently only some people, like some, like all their descent ancestors are like some specific place in Spain, and they can't trace that back, so they think they're all aliens.
Aliens. Yeah, there are some issues with RH positive, negative, and pregnancies that can be an issue. If the baby has one and the mom has the other, that there can be a problem there, an immune problem there. But yeah, so now we're going to talk about polygenic traits. So a polygenic trait is one whose phenotype is influenced by more than one gene. So not more than one allele. We were just talking about more than one allele with codominance and complete, sorry, not that. We were just talking about more than one allele. Now we're talking about more than one gene. So these are things that show a distribution, skin tone, height. These are polygenic. You have multiple genes that control your height, multiple genes that control things like your skin tone. So those are polygenic. So multiple alleles versus polygenic. Your multiple alleles, here's a great example. See all these options? But only one pair of alleles, only one gene. Here's your gene. These two together comprise your gene. You've only got one gene for this trait, but there are multiple alleles in the population. Whereas here, you've got multiple genes that are going to control your trait. So multiple alleles versus polygenic, polygenic meaning multiple genes. So 2 alleles form the gene which define a monogenic trait, those 2. But more than 2 alleles are present in a population, you get this multiple allele. But these genes, these 3 genes or sets of alleles work to produce your polygenic trait. All right, so I think this is our last Amoeba Sisters, but we got one more today because I think it's our last one, but they do such a good job at showing you the graphics.
Choose debit, pay later. Sign up with Clark.
Another, come on, show.
In the classroom.
Yeah.
University.
Hey, college, good deal. Amoeba Sisters are great for biology.
What do snapdragons, human height, and speckled chickens have in common? Well, they are easily identified as non-Mendelian traits. By non-Mendelian, we mean that genetically, they are rule breakers. They don't follow the regular Mendelian rule that having a dominant allele means the dominant trait will show. Remember in our guinea pig video, having a dominant allele represented by a capital H meant that the guinea pig would have hair? Only if there was no dominant present, a genotype of little h, would there be a hairless guinea pig. Well, that's a Mendelian trait. What we're going to cover now is when these traits are non-Mendelian and they don't follow these basic rules. So let's first start by snapdragons. We confess that when we heard this word, we thought they were some really amazing kind of creature. Well, they are amazing, but they're flowers. So I don't know, that's not exactly what we envisioned. Well, in snapdragon genetics, there can be 3 phenotypes: red, white, or something in between, pink. It's called incomplete dominance. In incomplete dominance, the dominant allele is not completely expressed when the recessive allele is around. There isn't really a clear dominant allele. If you cross a red flower, and we're going to write it here big R, and a white flower, little r, you are going to get offspring that are big R, little r. But unlike a Mendelian trait, if this is incomplete dominance, that big R allele is not completely expressed when the little r is around. So big R, little r in this case is pink. If you cross 2 pink flowers, big R, little r, like shown in this Punnett square here, you can get offspring that are red, white, or pink. Incomplete dominance is different from codominance. Codominance, like a co-worker, that prefix co should make you think together. They work together, the alleles, that is. For that reason, we like to use different letters entirely to represent the alleles. There is a codominance involving color in some types of chickens. Take a look at this Punnett square. If you cross a black chicken, represented here by BB, and a white chicken, represented here by WW, all the offspring here are BW. BW chickens are both black and white. They're speckled. See, both traits show up. This is the essence of codominance. And you know what's more awesome than a speckled chicken? Well, except for those silky bantam chickens.
Those are cool.
By the way, you will find that many people use different formatting for incomplete dominance and codominance. Some people like to use different letters entirely for incomplete dominance, and some people don't. Some people also prefer to do exponents with codominance, and some people don't. Formatting aside,
Isn't
when
that
you're
fun?
solving these Punnett squares, since we find this really does vary from classroom to classroom, the real concept is that in incomplete dominance, one allele is not completely dominant over the other. So you see this almost in between phenotype. In codominance, both alleles are expressed. It's the application that matters here. Height is fascinating. In our immediate family, Pinky is taller than Petunia. Our mom is also taller than Petunia. So how does that happen? There isn't just one height gene. There's a lot of genes that determine your height. What I mean by that is that you don't just have a pair of alleles, like big A, that will code for your height. It's more like someone having a genotype of big A, big B, big C, little c, and maybe more genes to ultimately determine your height. And you inherit one allele for each of the height genes from each parent. All of these genes work together to determine your height. Your skin color is also determined by many genes, just like your height. These are called polygenic traits. Poly means many, so many genes coding for one trait is what polygenic means. By the way, both height and skin color can be influenced by environmental factors as well. Nutrition growing up can affect your height, just as spending a lot of time in the sun can affect your skin color. However, this doesn't change the genetics for this trait. And finally, one more thing that we want to talk about. Epistasis. Epistasis is when one gene really depends on another gene for it to even be expressed. I mean, it really depends on this other gene. So let's pick an animal like, okay, a llama. Let's say this llama has a dominant big B allele, which means its wool will be black. So big B, or big B means that it will have black wool. And let's say that if a llama has a pair of recessive alleles, little b, then it will have brown wool. Now, what if there is another gene that controls whether this pigment will even be expressed in a llama in the first place? In this hypothetical example, a llama could have this other gene coded for by big C, big C, or little c in its genotype. However, if a llama has the genotype little c, it will not allow the other gene for wool color to even be expressed. Since we have two genes here, the gene for wool color and then this gene that controls whether the color is even expressed, this calls for our favorite 16 square dihybrid cross.
Yeah.
If you notice in this
Everybody goes,
dihybrid,
oh.
crossing two heterozygote llamas, any llama with big B and big B will typically give a black llama, and little b will typically give a brown llama. In all cases, unless they inherit a little c. If the llama has a little c in its genotype, then the gene for wool color is not expressed, and the llama is albino. This means that no pigment is expressed at all. Pretty interesting. There are many other non-Mendelian traits. We have another video on multiple alleles and a video on sex-linked traits. These are also non-Mendelian.
We're gonna get to more of those later. So, but yeah, so that's interesting, right? That's one way you can get albino. Actually, I put on our I shared an Instagram last night on our page, but it was about things that are satisfying in biology and things that are not satisfying, and one of them was a Punnett square. It was a minion. It was a minion like showing things on the screen, and the crowd of minions would either boo or clap. And the Punnett square, they all clapped. And then when it got to like a dihybrid or trihybrid cross, they went, ah, it's just too many things. Yeah.
There is incomplete dominance in humans, but their codominance like half is like one color.
Not like that. But there are some conditions that have, we'll talk about it later, but there are points where you can have genes that like turn on and turn off, for example. So usually it's like a sex-linked trait. So we'll talk about this when we get to sex-linked traits in the next couple of weeks. But for example, like alopecia or baldness is a sex-linked trait on the X chromosome. So you get that gene from your mother. So if your mother's father is bald, there is a chance you will be bald if you're male. That's most likely going to be where you're headed. But if you're female, sometimes those chromosomes, the genes can turn off and on at different times during embryonic development. So women don't bald like men do, but women, because they have that Y chromosome, which doesn't have a gene for baldness on it, that will turn on and off at different times during embryonic development. So women can get pattern baldness where you have patches, because this part of your scalp developed when the baldness gene was turned on. So maybe you're going to lose hair here, but this part of your scalp was developing when the baldness gene was being covered or turned off or whatever. And then, so women get pattern baldness, whereas men can get straight baldness. Yeah, but for men, it tends to be eventually, if you go long enough, you're going to lose all of it usually. But women get pattern baldness, which is different. All right, so here are your answers for your first ones. So your factors that determine traits are genes. Your offspring of true breeding parents with different traits are hybrids. Your specific characteristics that vary among individuals are your traits. Your alleles are different forms of a gene, and your sex cells or your egg and sperm or gametes. So what is the probability? Do we have what is probability? Yeah.
Something that could happen. Like
Something that could
chance.
happen. Yeah, a likelihood that a certain event will occur. In a parent pea plant with a allele pair, big G, little g, what's the probability that one gamete will have a big G allele? Colin. That's a 50-50. You're right. And come on, I'm clicking. There you go. Half or 50%. So if we complete this graphic organizer to determine the characteristics of homozygous and heterozygous genotypes. So a genotype homozygous is going to look like both alleles being the same, whereas heterozygous genotype is going to have what? Both alleles are different. Right. Now, for the phenotype, what's going to be expressed if it's homozygous? What are your options? Yeah.
Homozygous dominant than a dominant trait. Homozygous recessive, it'll be recessive.
Exactly right. And what if it's heterozygous? What's going to be expressed? Xander, your dominant gene is going to be expressed. Very good. All right, now the dominant allele for smooth pod shape in peas is capital S. The recessive allele for constricted pod shape is a little s. In a Punnett square, show the result of crossing two heterozygous parents, capital S, little s, and write your genotype and your phenotype. So what would we have in this square? Big S, which is heterozygous dominant. So what is our phenotype going to be? Everybody? Big S, which is what? It's going to show smooth, right? Yeah, non-constricted or smooth. How about here where we have a little s and a big S? What are we going to get?
Big S.
Big S, little s, and it's going to be smooth. That's right. And how about here where we have a big S, little s? It's going to be big S, little s, smooth. And then how about over here we have little s, little s?
Constricted.
It's going to be, yes, constricted. Very good. So what's the probability, looking at this, probability of a heterozygous offspring? Everybody.
75%.
Heterozygous? Heterozygous is, Colin? 25%. Heterozygous. Looky here, half. Because you have heterozygous, so 50% or half, right? What's the probability of homozygous then? Barrett? 50, but it's going to be half, 1/4 of that is going to be recessive, and the other 1/4 of that is going to be dominant. So 25, 25, homozygous is 50 in all. And then heterozygous is 50 and all. What's the probability of homozygous recessive, we said, everybody?
25.
25. And what's the probability of a smooth phenotype? 75. Very good. What's the probability of a homozygous recessive individual producing a gamete with a dominant allele of a capital S? 0. Why?
I don't.
Cause there's no.
Why, Ridley?
Because it doesn't have the capital S.
It doesn't have that gene to begin with, didn't inherit that allele, so it cannot produce that offspring. Okay, state the principle of independent assortment. Somebody have it? What you got, Kenna? Perfect. That's exactly right. Genes for different traits segregate independently during the formation of the genes. All right, so let's look at this. Oh boy, dihybrid crossing. We went from our nice, so satisfying Punnett square to fun stuff here. I'm just going to pop this up because it's easier for you all to look and see what you've got, and I'll give you a second to check it and make sure that you have it correct. Okay, so which genotype belongs to an offspring that is homozygous recessive for both traits? And what's the probability of that genotype? Eunice, what you got, bud?
CCSS, lower CCSS, all are lowercase, and it's 1/6.
There you go, 1/16. So you got a capital C, lowercase s, lowercase s, and it's 1/16 is your probability. What is the phenotype of an individual heterozygous for both traits? What's our phenotype going to be? Our capital C was green, and our little c was yellow, and our capital S was smooth, and our little s was constricted. So what's our phenotype going to be for a heterozygous on both traits? Eunice, do you have it?
So it's green, and then it's gonna be smooth.
Green and smooth. Very good. Because you're going to end up with a capital C, capital S, little s. So it's going to be like this guy right here. Going to be green and smooth. What is the probability of an F2? So we're in a second generation. F1 is your first generation. It says it right here. Gametes and some of the genotypes of F2 are given. Here's your F1, here's your F2. So what's the probability of an F2 offspring having a green pod and smooth pod shape? Anybody have it? Lucas, what do you have? 9 out of 16 is correct. And here are your options that would give you that phenotype. Now, Punnett square predicts a 9:3:3:1 ratio for phenotypes. What does that ratio mean? Lucas, what you got?
9 of them will be green and smooth, 3 of them will be green and constricted, 3 will be yellow and smooth, and then 1 will be yellow and constricted.
You got it. If it would click for me, click. There it is. There you go. So probability for both dominant traits is 9/16. One dominant trait and one recessive trait is 3/16, and both recessive traits is 1/16. All right, so looking over here, if we completed this graphic organizer to summarize exceptions to Mendel, we love Mendel. Exceptions get complicated. Mendel makes it so easy, but there are exceptions. So Mendel's experiments cannot predict the outcome of traits that involve incomplete dominance. An example, Caitlin?
Blend of white and pink and a flower.
Yeah, blend of white and red, giving you a pink flower. What is the speckled chicken feathers an example of? Claire?
Predominance.
Codominance. Yes, very good. Oop, that didn't pop up in the wrong order. Oh no, what happened? My order's done. That's stupid. Okay, so multiple alleles would be your 4 color codes in rabbits. And that's an organism's phenotype results from its genotype. I got my clicks out of order. What happened? Genes produce variable traits depending on their environmental conditions. Okay, Western white butterflies vary in their wing color because their environment varies depending on where they hatch. And what's this last one? Which is going now? Oh, what happened? Now my clicks don't work at all. Temperature affects wing color, and the other two just didn't fill in. But yes, you got to go back and check them. Yes, skin color would be what? What do we call that? Polygenic, and then what would this one be? These codominants, Claire said, so that was right. It just didn't show up. I don't know why. I'll have to fix that. All right, these are up for you to check. Let you have a look at all of these and check them, since it's a lot of just short answers and a chart. Did you guys know before that the temperature at which turtle eggs incubate determines their sex?
Yeah.
It's kind of wild, right? That's really wild.
If they're at the bottom, and then it's like.
Mm-hmm.
Does that work with children?
No.
What if you're like right at a temperature?
Do what?
What if they're right at a temperature like where it's
I guess it's a 50-50 chance then, as opposed to chances being, it's probability, as always.
Do you know what the chances are of a coin flipping heads?
Isn't it 50-50 or is it not? Is it different?
Isn't it 51-49?
It's 51-49 because the head side has
Slightly
it has
mass.
more mass. On
More
it.
mass, yeah.
Mhm.
In practice, it's probably close to 50-50. You'd probably have to flip it tens of thousands of times to get to that.
It was like computer gene.
Yeah, you'd probably have to flip it a ton to get to that. All right, so I'm gonna let you take this today. I'm gonna put these out for you to take. All right, now I'm gonna set these back out.
Get a break.
What you're gonna do,
Also.
hold on, I gotta get little bags for you. I forgot to grab the bags, so I gotta grab little bags for you. But what you're gonna do is you're going to each take with you, whoops, one of these worksheets, and you're going to take 2 strips of paper out of every one of these cups. These are your alleles. And you have a bunch of different traits on here. Okay, I just forgot to grab bags, so I gotta grab bags for y'all. That'll just be a second. So you're gonna pull your 2 alleles for your dragon. You're gonna build a dragon basically today. And then you're eventually going to mate your dragon. This isn't graded, but we're gonna be doing it in class, so don't lose it. Keep track of it. Come on, keep track of it. All right.
Wait, so pre before dragon?
Eventually. First thing you're gonna do is create your parent dragon. You're going to go through this and you're going to actually draw your parent dragon based on the traits you get. And the traits are going to be things like whether it has wings, fangs, how many claws it has, how many horns it has. So you're going to take 1 copy and you're going to take 2 strips of paper from each cup. Question?
So the question was, like on the end of the lesson 9, there's this crossword puzzle. Is that the homework?
Yes, there's a homework question.
We have to turn it in.
Yeah, I have to double check it, but yeah, I think that was right. Yeah, all right, so I'm gonna put all this over here, and I'm gonna run and get your bags. Don't leave till you put your alleles in a bag, because you will for sure lose them. All right, so, hold on. I gotta walk this way. Take them. Take 2 out of every cup. And I forget 1. 2, just bring it, don't look, just pull.
2 for every cup?
2 from every cup. And let me go grab, I'm gonna have bags for you guys. I have bags for my other grade.
Oh, I have. Oh my god. Maybe so. I had to go why is there so many? I
think your mom.
I need to go for the best dragon. Hey,
I'm pretty sure I have 3 head sounds, so.
nobody will know. Secondary. Your dragon's gonna have the
I'm an immensely impaired dragon.
But you can eat that.
It's gonna happen. It's gonna have 3 brains.
Sheets, so many down 2 strips from each cup. Here are bags.
Possibly took 3 from one of them, I'm not sure.
I have 2 long tail. He's got too long.
You can take these home and work on them, but keep them in your binder because you're gonna keep working on them in class. You're gonna want to have it, okay, where you can access it.
So what is the dragon just 2 months?
It shouldn't. I don't think there's an allele for number of heads.
I grabbed Excellent.
It says don't spot the neck and blue spot the neck. You have two necks or like one?
No, you have no spots on neck and then you have two spots on neck. So you have to, which one is dominant?
Oh. I can choose from Bobby.
No, you don't get to choose and tell you.
Oh, yeah.
Yeah, just pick two out of each cup. Here, Tyler, come on down here past Grace. File in this direction. So you're gonna mate through it because you're gonna build your parents and then you're gonna actually mate your dragon with somebody else's dragon. And then you'll have a big dragon. No, so it's just, we're gonna just work on it for the next couple weeks.
When we got.
Yeah, so these are your alleles. So, one that is dominant. So he's gonna have a nose spike. Yes, they're gonna be, some of them are gonna be like that. Yeah. Yeah, we're gonna keep working on it for the next couple weeks though, so keep it in class, keep it in guidance. Okay, so you got all the types, right? Next is coming in. We got a lot today. What you doing? You bring extra money?
No.
Two of each.
Oh, organize some.
Tyler, if you come to this side, you can start down here and work this way. Just come around. We need two eggs, step in. Two from each cup. What? There you go. You're gonna build your little dragon with a little bit, cute little dragon.
Do we need to, does this need to be completed by next week?
No, just keep working on it little by little. We're gonna work on it in class. So you're gonna learn your genotype and phenotype of your parent dragon, and you'll work on your dragon over the next few weeks. You'll eventually breed your dragon. I'll let you, it says leave it here, don't take it home, but you can take it home. Just leave it in your binder is what I mean. Like, don't pull it out in your bedroom and forget it, because you'll need it next week. And now they just see themselves out.
No.
They get their stuff and out the door they go.
I don't know. I think so.
Oh, yes, yes. Well, a lot of these guys come to my Harry Potter party. These two,
Oh, yeah, I remember. I
recognize a couple of faces. Yes, because you came too. Yep, it's always fun.
Okay, this is a very special amount of paper, guys. Okay, worry about making sure I can later.
No.
What is she like?
Oh, just toss that.
The same, see, so I'm going to color eye right here. So it's.
Well, you sure do. That's so cool. Yeah.
But any info I was talking about today.
There's probably some code evidence there. That's really cool. That's really interesting. You don't see that very often.
Because I don't know my parents have this.
That's beautiful. That's interesting. And so it's probably something different. There's lots. I've colored one of those ones that be, you know, if it's solid blue or solid brown, it tends to be simple. But when you get into green or other variations, it's all, there's all kinds of factors that play in. It's really interesting. That's gorgeous. So I mean, that's close. It's really pretty. Yeah. My glasses distract from the pretty eye. Very cool. Then you get people that have two completely different colors. I have a friend from high school who married a guy who has one brown and one blue. It's really pretty.
It's all that thing.
It's heterochromia. Yeah, heterochromia. Yep.
I think that's actually unique.
Yeah, it's very unique.
Yeah.
Very uncommon. You gonna breed your dragon?
Bruno.
Drew?
He's going to put a hold on.
Who's gonna hold it? Okay, no, let's you can leave it right there.
I don't want to take it home.
Yeah. There you're good. He's getting ready to leave because he's got to go coach. So that's what he's doing.
So much fun.
Oh yeah? You like it?
Yes.
Good. It's gonna be fun.
Didn't I have a very good one?
No, Ridley was just wondering where you went, so that you wanted to make sure you knew that the file was here. The file, your case file.
Oh.
I don't know if you wanna, where you wanna put it or store it or whatever. Here. Just wanted to make sure that you knew where it was, since you were maintaining it. 17 days of Halloween.
Yay! Simple sticker. Photo.
17 more possibilities.
Yes, yes, more possibilities for Tinker Day coming. It's coming. Yes. Hinton knows, but nobody else knows.
I'm gonna go ahead and message on a Saturday.
Never on a Saturday. Always Monday through Friday.
Alright, fine. It's gonna be on a Tuesday. No, how about Monday?
You think, huh? Is it what?
No thing.
I think so. Yeah, should be no Fang, I think.